Okay, so my lab's located at the University of Utah, and we have an interest in epigenetic effects, but our kind of unique niche and angle on that problem is studying those effects at the allele and cellular level. And I'm going to talk about a paper that we published in February issue of Neuron of this year, and that's going to be the kind of the major focus of this talk. So I'm going to start out with a little bit of a general background to get everybody thinking on the same about the same problems. For every autosomal gene inherits one copy from mom and one copy from dad, and the typical view that we have of the expression and regulation of those two copies is that they're by and large regulated equally. And so if you were to think at the cellular level, you'd have two copies and they would both be expressed. And my lab's interested in the phenomenon where you get an epigenetic mark that impacts one allele and not the other. And so as a consequence, one allele is maybe dialed down or turned off, and the other allele is expressed. So these allele-specific epigenetic effects can take on a variety of different forms, potentially, and we're interested in what forms of these effects exist in the brain. We think that this is an important problem because as our understanding of genetic risk factors that influence mental illness and brain disorders has grown over the past few years, we've learned that it's frequently the case that just one allele is impacted. So for example, in the autism spectrum, we have a sense that 80% of de novo coding sequence variants are paternally derived, so they're just impacting one allele. 20% are maternally derived. Even in the context of copy number variation, it's frequently the case that just one allele is impacted, and recessive mutations, so far as we can tell, don't contribute appreciably to most of these major forms of mental illness, schizophrenia and autism, for example. And we've learned that paternal age is a risk factor for mental illness and other disorders, presumably due to increased mutations in the germline. So on average, as fathers get older, you get about 2 new mutations per year that are transmitted to offspring. And so this is a risk factor. So we've learned that one allele is impacted genetically, and if one allele is impacted epigenetically, then that can have some interesting implications potentially at the cellular level. Our current picture of a heterozygous mutation and how it's expressed at the cellular level is that you'd have one mutant allele and one wild type allele within a cell, and those would be biallelically expressed. So the cell would be expressing both copies. But if you get allele-specific epigenetic effects, you could have some very interesting outcomes. You might have cells that have turned off the mutant copy, and so they're only expressing the healthy wild-type copy. Or on the other hand, you could have the other scenario where the healthy copy has been silenced and the mutant copy is being expressed, and presumably that would be bad, right? And these would be cells that do not have a healthy copy to buffer the effects of the mutant copy. We know a bit about this from the field of genomic imprinting, which is where my lab has done a lot of work in the past. And imprinting is the scenario where epigenetic marks established in the sperm are different from the marks in the egg, and the maternal and paternal complements of the genome come together, and those epigenetic marks are inherited, and as a consequence, for some genes in the genome, there is a preference to express the paternally inherited copy and silence the maternal copy, or vice versa. And this can have interesting implications for genetics. So one of the classic examples is the Angelman syndrome and Prader-Willi syndrome situation, where there is a cluster of these imprinted genes on chromosome 15. And if you inherit a deletion in that cluster from your mother, you'll get Angelman syndrome because you lose the expression of a gene called UBE3A that's only expressed from the maternal allele. On the other hand, if you inherit the same deletion from your father, you get Prader-Willi syndrome. And this is because there is a set of genes that are only expressed from the paternal allele. And so the same mutation can give rise to two different disorders because different genes are being expressed from different alleles. Okay, so that's an example of how the intersection of genetics and allele-specific epigenetics can have an interesting impact on phenotype and disease risk. And what I'm going to talk about today is trying to understand the full landscape of these epigenetic allelic effects in vivo in the brain. And we're going to go through the mouse brain and the macaque brain, a little bit about the human brain. What we know right now is there are these imprinting effects. We have a pretty good understanding of the landscape of those effects in vivo, in mouse, and a growing understanding in human. We know about random X inactivation, and so this is instead of always silencing the maternal copy or always silencing the paternal copy, we randomly are choosing the maternal or paternal X, and this happens, of course, in females. And there are some genes on the autosomes that are well known to do that in vivo as well, olfactory receptors and clustered protocadherins, for example. Okay, the model system that we've kind of set up to do this in mice works in the following way. We have two distantly related strains of mice. One inbred line is called Castaneus, and the other line is called Black 6. And we cross those, and in the initial cross, Cast is the mother and Black 6 is the father to generate an F1 hybrid. And so half of the alleles come from Cast and half from Black 6. And then you can do RNA-seq on any tissue at any developmental stage that you're interested in and phase the reads at the polymorphic sites in the genome that distinguish the alleles from these two strains, and just tally the reads that come from the maternal allele relative to the paternal allele, and you get this genome-wide readout of allele-specific expression in the brain at in any region or age that you're interested in. If you're looking at imprinting, which we've frequently done in the past, it's particularly important to do the reciprocal cross, where now Black 6 is the mother and Castaneus is the father. And you profile gene expression now in this F1 hybrid from the reciprocal cross, and would show, if you're interested in an imprinting effect, that the expression bias always comes from one parent's allele. So in this example, the maternal allele, even though Black 6 is now the mother, we're still always getting a bias from that allele. Okay, so that is the general methodology, RNA-seq on F1 hybrid mice, and that's where we start. And what we're going to test in this study is the null hypothesis that maternal and paternal alleles are equally co-expressed. So the idea is that you have these genetically identical inbred F1 hybrid, these F1 hybrid animals, and we do a dissection from each one, and there's just going to be variance in the dissection a little bit in terms of the cells that you get and the physiological state of the animal. And our null hypothesis is that you're going to get these fluctuations in the levels of gene expression between different individuals, but the maternal and paternal alleles will go up and down together for most genes because they are co-regulated together, right? And what we're interested in potentially discovering are scenarios where that is not the case, where the maternal and paternal alleles appear to be decoupled, and so they're no longer correlated, and that suggests that they're being regulated independently. So that's the idea of the screen, and I'll just show you kind of what that data looks like here for a gene called FUCA2, where we've developed a strategy that centers the RNA-seq data, and we've done RNA-seq here on 18 different mice, and we're looking at the expression of the maternal allele in red and the paternal allele in blue, and you can just see this jitter in the level of expression of FUCA2 across these different individual mice. And for this gene, the maternal and paternal alleles go up and down together. So they are in lockstep sync with each other, suggesting that the two alleles are being regulated equally. And so they're correlated in this case. The correlation, the Pearson, is 0.9. So here's another look at the FUCA2 data in another way of presenting it, where now we're plotting the expression of the maternal allele versus the paternal allele, and we're doing that for all of these different 18 mice, and you can see again they're highly correlated. Just another way of presenting the same data. But when we look at other genes in our data set using the same strategy, we can find genes that are very exciting, like BMP7 or ADORA2B. And BMP7 has a very low correlation between the maternal and paternal allele. So it suggests that when the maternal allele goes up, the paternal allele is free to do more or less what it would like, something else. It can go up, not change, or go down. ADORA2B has a negative correlation between the alleles. When the maternal allele goes up, the paternal allele goes down. And so this is an interesting relationship between the two alleles that we've uncovered in the data. And you can imagine that these effects would be due to technical noise or all kinds of other problems, and we're going to step through how we deal with that. But I want to start by giving you some conceptual framework to think about what's being detected in the data. And I'm showing some beautiful results from a paper by Jeremy Nathan's group that was published in 2014. And what the Nathan's lab did here is they put reporters on the maternal and paternal X chromosome in mice. And so the maternal X, when it's expressed, the cells that chose that are labeled with GFP, and the cells that chose the paternal X chromosome in female offspring, those cells are labeled with dimer tomato, so they're red. And here is a set of animals from the same litter, and you can see that females within this litter go from animals that, they're a mosaic, but they're predominantly, this just by chance, this animal is dominated by cells that chose the maternal X chromosome. And this one is kind of a mix of cells of the maternal and paternal X, and this one is more of the paternal. You go all to the, the far extreme of being of an animal that predominantly its cells have chosen the paternal X chromosome for whatever reason, right? It's just a stochastic mosaic effect in the offspring. And if you look in the brain, this mosaic can be very striking, where one half of the brain within a single animal is predominantly selected the paternal X chromosome, and the other has selected the maternal X. And so this is what random monoallelic expression looks like at the cellular level. And you can imagine what we're doing is we're cutting out a chunk of brain, and we're looking at the correlation between the maternal and paternal alleles for different genes. And so when we do that, we can detect a landscape of different allelic effects potentially. At the cellular level, if the maternal and paternal alleles are equally expressed in each cell, then we expect that they will be highly correlated in our RNA-seq data. When the maternal allele goes up, the paternal allele will go up, and they'll be tightly correlated together. If the effect is random monoallelic expression, like the X chromosome that we just saw from Jeremy Nathan's paper, then if more cells choose the maternal allele compared to the paternal allele, then they're always coming at the expense of one another. So they're going to be negatively correlated. So in our data, if we see a negative correlation, like that example of ADORA2B, then that is potentially indicative of random monoallelic expression effects like the X chromosome. There are other potential things that can be detected with this screening strategy. We can detect genomic imprinting. So that's the case where one parent's allele is completely off. And in our data, there's going to be no correlation between the maternal and paternal alleles because, of course, there's no expression coming from one of them. If we get cell type specific imprinting, where some cells are biallelic, other cells are only choosing one parent's allele, then that's going to drive this correlation down, and you'll get a low correlation. And you could get that also from random monoallelic effects, where some cells are doing the random monoallelic expression thing, and others are biallelic. And that will also drive this correlation down. So that example of BMP7 that I showed that had very low correlation between the alleles, that's potentially indicative of a cell type specific allelic effect. Okay, so I hope that helps for everybody to understand some of the data that I'm going to show and the conceptual framework that we had in place when we designed this screen. I mentioned before that one of the challenges with doing this is if you see a low correlation, it could be due to technical noise, it could be due to biological variation in the data, other problems. And so what we needed to solve, and this is actually a big part of the paper, it's buried in the supplement, but it was a big part of what we had to work through, is a statistical framework that would give us confidence that we are about what the underlying biological correlation between the two alleles actually is. And so I'm not going to go into it in great depth in this talk, but I'm going to say, the important take home is that we worked out a kind of empirical Bayesian strategy to estimate technical noise in the RNA-seq data. And we also model biological variation in gene expression between different individuals. And ultimately, we go on to model cis-EQTLs in the macaque data that I'm going to show you, with the final outcome of defining a 95% confidence interval that reflects the underlying biological correlation between the two alleles, okay? So we have this range where we're 95% confident that the true correlation between the alleles lies in that range. We call it the RAB. And this strategy works quite well. X-linked genes that are predicted to be random monoallelic have a negative correlation. Genes that are imprinted and should have no correlation between their alleles. So these are known imprinted genes. They behave that way with no correlation. And then many autosomal genes that are predicted to have equally co-expressed alleles, they have highly correlated alleles, which they should do, okay? So now we can apply our screening strategy in a genome-wide manner. And we start out by focusing on females because we want to use random X inactivation as an internal control to set cutoffs and thresholds in the data. So we do our screen, and then we have this unusual plot in the data, and what we're showing here is for every X-linked gene, there's 500 of them expressed in this part of the brain that we're focused on initially, the dorsal raphe nucleus, which is a major serotonergic region of the brain. And these little lines that we're plotting are those 95% confidence intervals for the correlation between the maternal and paternal allele. And the y-axis is kind of down here, so -1 is a negative correlation, 0 and +1. And what this shows is that for X-linked genes across a range of different expression levels and variation characteristics in the data, we can detect the low correlation between the alleles robustly. There's a lot of blue and green and orange bars in this plot, and that is indicative of these differential allele expression effects. And we can see that the upper bound of the 95% confidence interval for most X-linked genes bumps up against plus 0.75. And so we set that threshold because as a marker of X chromosome-like differential allelic effects because it captures most of the X-linked data there. There are some genes, like this little red bar you can see here, that have co-expressed alleles and they're X-linked, so they're highly correlated, and those turn out to be genes that escape X inactivation. So this gene in particular is DDX3X, and that is a known gene that escapes X inactivation in the mouse. So that's also a nice outcome. So now we apply the screen to the autosomes. And what's immediately obvious when you look at the autosomal data is that it's all shifted up towards +1. And we expect that most autosomal genes are co-expressing their alleles more or less equally, I think. But what is exciting is to see, was there a question? Okay. What's exciting is to see that there are genes that have the properties of X-linked genes, even though they're autosomal. And that's what these colored bars indicate, are these genes that meet the threshold of the X-linked genes in the data. There's also a subgroup of genes that have tightly co-regulated alleles, highly correlated alleles, and we also set those out as being interesting and unusual, and they're indicated in red there. Okay, without getting into it in great depth, we needed an orthogonal approach to validate that this screen is actually picking up what we think it is picking up and that it's reproducible with another method. And so we develop an approach using pyrosequencing to also measure these allele correlation effects. And we do the validation across a number of genes randomly selected from the data. And the pyrosequencing validations are tightly and significantly correlated with the data set, the results we're getting from the RNA-seq. So we can validate the effects that I just explained with an orthogonal approach. And so we think that this is reproducible and robust. So in summary, we've got a screen that detects these reproducible differential allelic effects in vivo for the first time. And we have a couple of questions that we need to address. One is, are these effects related to differences between the cast, genetic differences between the cast versus B6 alleles? What is the relationship between these effects and the expression level of the genes? And what is the impact of genomic imprinting in this data? So the first problem or question that we want to dig into is, are genetic differences between cast and B6 contributing to the results we're seeing? And the short answer is not appreciably. So here's a gene, for example, AK7, and it expresses the cast allele at a higher level than the B6 allele due to cis-EQTLs that are different between the strains. But the two alleles are nonetheless highly correlated and in lockstep with each other. Here's a gene, MARPL16, and the two alleles are not different, so there's no strain difference in the expression level of that gene, but nonetheless, the correlation in their expression is very low. So they're showing these differential allelic effects despite the fact that the expression level is the same between the two strains. And ultimately, we can just do a kind of linear regression analysis and get a sense of how strain differences contribute to the variance in differential allelic effects. And 90%, over 90% of the variance in the data is not driven by this. So it's driven by something else. And we think it's non-genetic. And I'm going to go on to show you evidence that that is likely the case in most of this data we described. Gene expression level is not something that plays a strong role in driving differential allele expression versus the sort of co-expression of the two alleles. There is no significant difference between genes with differential allelic effects plotted here. This is the level of expression of these genes, and genes that have negative correlation compared to all autosomal genes. There's nothing significant there. Genes that tightly co-regulate their two alleles, they are expressed at a slightly higher level than all autosomal genes overall, and these genes that show the differential allelic effects. So that unusual class of genes that have tightly co-regulated alleles, they have a slightly boosted overall expression level. Most of the effects we're uncovering in the screen are not caused by imprinting. So we have already worked out robust methods to detect imprinting in this type of data. And very few of the genes that show up with these differential allelic expression effects are doing this because of imprinting. So it's something else. Okay, so we have a genome-wide approach to screen for these effects, and we want to understand, like, what is this and why might it be important? We applied the screen now to a variety of different ages and different tissue types. And the results from this screen were really surprising and interesting, I think. We profiled differential allele expression in the postnatal day 5 So this is just the neonatal brain in the dorsal raphe. In the juvenile brain at postnatal day 15, so this is getting close to the weaning stage of life, and then in adults. And then we tallied the number of genes at each of these different developmental stages that have these differential allelic effects versus the co-expression of the two alleles. And what the data show is that at P5, during this early stage of development, many genes, greater than 85% of the genes, show statistically significant, high confident differential allelic effects. So during early stages of brain development, our data suggests that the two alleles are decoupled frequently for most genes. And we validate this with another data set independently generated for the cerebellum. And then as the brain matures by postnatal day 15 in adults, the two alleles start to come into alignment with each other more and more. Presumably as cells differentiate and mature, there seems to be this effect of bringing the alleles in alignment with each other. So you see more red. That's more of these genes that have tightly co-regulated alleles. You see more gray, and so those are genes that do not fall into any unusual class in terms of differentially expressing or co-expressing their alleles. But there are nonetheless, in adults, 10% of genes that are differentially expressing their alleles.
Chris, do you have any way to alter the expression of the autosomal genes during the early development so that they are not differentially expressed, to see the biological consequences of that?
Yeah, so that's a really great question. And the short answer is that we don't know what the mechanism involved in this is. And there are a variety of possibilities. I have some favorite ideas that are related to competition for enhancers and regulatory elements for genes, and so that competition could be resolved in a kind of allele-specific manner. But we don't have a deep enough understanding to do that type of experiment yet.
Okay.
Yeah. The arcuate nucleus of the hypothalamus, we profiled that region in adults. We profiled liver, which is endoderm-derived, and we profiled skeletal muscle, which is mesoderm-derived. And the short story is that these effects were present in all of the different tissues and brain regions that we looked at. And one of the questions we had is whether this would be uniquely enriched in the brain compared to non-neural tissue, and it doesn't look like it is particularly enriched in the brain. The big take home is that it's very prevalent in the early developing brain. So this is a sort of summary slide of what the pattern in the data is, where you've got this decoupling in the neonates, and then it kind of goes away as you get older. Okay, so we wanted to understand what this is at the cellular level. And in the past, we had worked out an approach to resolve allelic expression at the cellular level using a nascent RNA in situ strategy. And the way this works is we use RNA-scope probes, which are very sensitive in situ probes, and we design them to rapidly process intronic sequences in the nascent RNA. And this, in effect, detects transcription on the DNA allele. So you can see active transcription in tissue samples. We cut sections of mouse brain, or I'm going to show you human brain, and stain for allelic expression for any gene that you're interested in. And in this case, I'm showing controls where we've looked at synapsin 2, and it's a biallelic gene. And we're looking at monoamine oxidase A, and this is an X-linked gene. We're looking at females, and so that means that due to random X inactivation, most cells will have silenced one X and should only be expressing one allele. And what you'll see here in the mouse brain is this is the nuclear counterstain, and there's 2 dots for synapsin 2 in each of the cells, showing both alleles are transcriptionally active, and 1 dot for monoamine oxidase A, just 1 allele is active. So we can see these allelic effects. Great. So now we turn back to the genes that we predict to have co-expression and genes that we predict to have differential allele expression from the RNA-seq, and we design probes to them. And then we look at the number of biallelic versus monoallelic cells in the brain for these genes. And the results are quite clear. MTAP1B is an example of a gene with co-expressed alleles in the RNA-seq, and most of the cells are biallelic in the mouse brain. So they've got 2 dots in the nucleus. But BMP4 is an example of a gene with these differential allelic effects, and most of the cells show 1 dot in the nucleus, suggesting, monoallelic expression. And then we do this for many genes, and we're able to find that there is a correlation, a negative correlation between the the allele co-expression effect, where genes that have tightly correlated alleles have very few monoallelic cells in the brain, and genes that have low allelic correlation have many monoallelic cells in the brain. Okay? So I hope that's clear. So we have now a kind of growing sense that what we're detecting in the RNA-seq reflects allele-specific expression effects at the cellular level. And when I started out the talk, there was this potentially important implication for our understanding of genetics and how heterozygous mutations might be expressed. If you got these allele-specific effects, then you might have some cells that only express a mutated allele and some that only express a wild-type allele. We should be able to see these kind of mosaic effects for the genes we're detecting. And so we want to test this, and we acquired heterozygous mice. They're knockout reporter mice. So that means that a LAC-Z reporter has been inserted into one allele, essentially making that allele a null. But we can detect the expression of the mutant allele and the wild-type allele by designing RNA-scope probes now to the mRNA, not the nascent RNA, but the mRNA. And this is kind of semi-quantitative because each dot, when you do this, is one transcript. So now we can get a look at the cellular level of the expression of the mutant versus the wild-type allele in these mice. And when we did that in the P5 brain and in adult for BMP4, we were interested and excited, I guess, to see that there is a mosaic of cells in these animals that's clearly visible. Some cells are expressing the wild-type allele, some are expressing the mutant allele, and then some are biallelic. And so here are the zoom-ins there of the biallelic cell, the mutant cell, and the wild-type cell. And then we saw the same thing in the adult brain. We use this strategy to look at a few different genes. There are a lot of mice that have been engineered this way, and so that's a great public resource to tackle this problem. So we got 3 different lines for genes predicted to have these differential allelic effects, and we looked in the brain, and there were always cells that were strictly wild-type allele expressing or strictly mutant, and there were things in between, the biallelic cells expressing both mutant and the wild-type copy, and then we saw kind of dominant effects where predominantly mutant, but a few wild-type transcripts were detected, or the other side, the other way. When we tallied these in different brain regions and cell types, there were interesting brain region differences and cell type differences. So for example, in the RAFA nucleus, the DRN, or the kind of ventral periaqueductal gray region, if we look at BMP7 and BMP4, most of the positive cells fall into the monoallelic class, where they're wild-type monoallelic or mutant monoallelic. But there are some that are biallelic. But when we look at a totally different cell type, which are meningeal cells, there's very few of these monoallelic cells. Almost everything is biallelic. And so this suggests that these effects can be cell type specific. And we dug into that in a little bit more detail. We used the nascent RNA approach to look in the arcuate nucleus for BMP7, and you can see most of the cells in the arcuate of the hypothalamus are monoallelic, 1 dot by nascent RNA. And then we look in the heterozygous mice, and most of the cells are just expressing in this field the wild type allele, so they're monoallelic for the wild type allele. But when we look at meningeal cells, there's 2 dots in the nucleus, 2 active alleles. And then look in the heterozygous mice, and you can see that these meningeal cells are expressing both the mutant and the wild type allele, right? So cell type specific effect, allelic expression effect, yeah. Okay, so we got a way to screen for these effects in vivo, and we have some data that suggests that these are allele-specific expression effects happening at the cellular level, and they can interact with heterozygous mutations and shape the expression of those alleles at the cellular level in vivo. And so this could have some implications for mental illness, potentially. And so we want to know, does this happen in the primate brain? Okay, and so we're going to move to a study that we did in macaques. And the strategy here was we had 10 trios, mother, father, daughter trios of cynomolgus macaques. And we sequenced the genome of dad and mom. And then we did RNA-seq on the dorsal raphe region in the daughters. And so now we know the variants, and we can phase the alleles just like we did in the mice. And to make a long story short, we can now plot paternal allele expression versus maternal allele expression. And immediately we could see in the data that there were, just like we saw in the mice, there were genes that tightly co-regulated the 2 alleles, but there was also evidence of genes that did not. So that was a good start. And we had to deal with a few different, there were a few tweaks that had to be done to the statistical modeling, because we needed to estimate the impact of kind of stochastic cis genetic variation in these outbred macaques. And in short, we did that in part by drawing on the mouse data, where we knew what sort of the distribution of genetic allelic effects looked like in the mouse data, and we could randomly draw those effects out of that and incorporate it into our Bayesian model. And so I think our approach is conservative, maybe too conservative, but it nonetheless was a good first pass for this paper. So we apply that approach now to look for differential allelic effects in the macaque. And for a gene to be included in our analysis, it has to have a variant in all 10 daughters. And so that limits the space of the transcriptome that we can actually evaluate. And in the end, there were 900 genes that we could look at that met that criteria. Several of them were X-linked genes, and so again, we could set a threshold from the X-linked genes to detect differential allelic effects, and X-linked genes had relatively low correlation. And then we looked at the autosomal genes in the macaque. They're all shifted up towards +1, which is great, but again, we could find genes on the autosomes that had these properties like X-linked genes. And some of them were very important, like Huntington, for example, which is an autosomal dominant disorder. And that was one of the top hits out of our screen. And we've gone on to do some validations of that in human brain tissue, and it seems to be looking quite good. So, so out of our macaque data, we found genes that have tightly co-expressed alleles. We found genes with evidence for differential allelic expression, so slightly low allelic correlation or strong allelic decoupling, I should say. And so we followed some of those up. And 2 genes that were of particular focus for us in this study were CNT and AP2, which had correlated alleles, and DEF1, which had low allele correlation. And those two genes are interesting because they are linked to autism spectrum disorders and language development. And we developed probes to those genes and acquired tissue from different brain regions from the Taminga lab at UT Southwestern. And so we were able to stain tissue from the orbital frontal cortex, Broca's area, the anterior cingulate, and the dorsal raphe, and look at allelic expression for these genes. And CNTNAP2, as was predicted by the macaque data, was predominantly biallelic in the cells that we resolved in human brain, and in both the ACC and the orbital frontal cortex. But DEF1 showed evidence of monoallelic expression in most cells. And we looked across 13 unrelated females, and the pattern for this allelic expression was quite reproducible between those different individuals. So for DEF1, over 80% of the cells that were positive were monoallelic, and a small number were biallelic. And for CNTNAP2, it was the opposite situation, but also, again, highly reproducible across different individuals, mostly biallelic, a small population that was appeared to be monoallelic. So in summary, we have this growing landscape of non-genetic allelic effects that we're just trying to get a better understanding of. There's been studies of genomic imprinting, and there's a relatively small number of genes that show really robust canonical imprinting effects in the brain, maybe 40. In mice, my group and the Dulac lab have described these non-canonical imprinting effects that are more frequent than canonical imprinting, but still relatively infrequent. And now this study that we provided here recently has provided evidence for other allelic effects that are more frequent, particularly prevalent in the developing brain, and we don't have an understanding of the mechanisms involved or the function. So that is my talk today. The people that led this study, the differential allelic expression effects, it was Wei Chao Huang, who's a graduate student in my lab, and Elliot Ferris, who's a computer scientist in the lab. And it was done in collaboration with a statistician at the Huntsman Cancer Institute, Ken Butcher. The primate genomics was done in collaboration with Janice Wagner at Wake Forest, and the human brain tissue analysis was led by a technician in my lab, Tina, in collaboration with Carol Taminga's group and Kelly Gleason at UT Southwestern. And if there's any questions,